The main focus of this research is the design and development of an interactive visualization tool called SynVisio that can assist genomic researchers and biologists in interactively exploring genomic conservation. SynVisio offers multiple visual representations such as parallel plots, matrix-style dot plots, and even multi-genome comparison plots such as hive plots. Further, it also lets researchers explore the datasets in multiple coordinated visualizations and offers them the ability to dynamically refine these visualizations through a combination of interaction techniques. But before we get into the design of the system, let's take a step back to look at the bigger picture and understand the motivation for developing such a tool. Now, one of the defining characteristics of genomic research in the last couple of decades has been the generation of enormous amounts of biological data, both about humans ourselves and the organisms that surround us. To put this into perspective, let's look at the Human Genome Project, an endeavor that was started by an international group of researchers in the early 90s to sequence the complete human genome, a feat that would take almost an entire decade to finish and cost over $3 billion. And fast forward to today, you can get the same data for less than $1,000, and all you need to do is spit in a cup and wait for a couple of hours. This unprecedented improvement in sequencing genomes has led to an explosion of quantitative genomic data, and one particular field where this data is extensively used is comparative genomics. Comparative genomics primarily involves comparing genomes of two or more organisms to study the similarity where each genome is the complete DNA of that organism. Sequence similarity can of course be studied at several levels. At the single gene level, duplicated sequences can often imply that both these genes code for the same protein and thus have a similar function. However, when several collinear sections of such genes are duplicated, they can shed light on the evolutionary history of the two genomes. This is due to the fact that as organisms evolve into different species over time, they end up retaining parts of their DNA from their common ancestor. Such collinear duplicated regions containing several genes are referred to as syntenic regions, and visualizing the relationship is the primary goal of our visualization tool. But this brings the question, why is visualization necessary in this context? And the answer to that question is that even though a large part of genomic comparison is purely computational and can be automated, human judgment is still vital in certain scenarios. And that is where visualization tools come in, because we as humans are intuitively good at identifying patterns in spatial representations. To prove this, let's take a look at Anscombe's quartet, a beautiful example of how four different datasets, which at first seem statistically identical because they have the same mean, variance, and correlation, and yet visualizing them makes it pretty obvious that they are completely different and each of them tell a different story. Or let's take another example much closer to home and look into our own DNA. Now, if we were to compare this with the DNA from a chimpanzee and connect the similar regions, we can immediately tell that both our DNAs are nearly identical and that at some point in our evolutionary history, these two regions in a chimp's DNA fused into just a single chromosome in humans. And while this is just one example, genome researchers are increasingly relying on visualization tools to both hypothesize and investigate diverse and complex evolutionary tasks. However, one of the biggest impediments to using genome visualization tools that currently exist is that they do not support a wide variety of investigation tasks and often have limited or no interactivity and purely exist as chart generation systems. This makes it difficult for researchers to understand the rich characterization of the complex genomic datasets that they sequence. Some visualizations, such as this popular chart for synteny, are in fact not even based on sound visualization principles. This chart, for example, relies on categorical color differences to depict similar regions, even when decades of visualization research has shown that humans are particularly bad at noticing subtle differences in color. And in fact, color is one of the least distinguishable visual channels compared to things like possession, shape, and research. Now, using colors itself is not bad, but they should be used in the right context along with more noticeable visual variables, which goes to show that there is a huge scope for improvement in this design space. And so we set out to build an interactive visualization tool that addresses these limitations and can be used in a wide variety of scenarios. And to do this, we gathered our requirements by working closely with 3 teams of genomic researchers, working in different areas of biology, plant science, and crop breeding. We met with these teams numerous times over the last 2 years to understand the various genomic tasks they perform and distill them into 6 major visual and interaction-based requirements. Now, let's take a look at how SynVisio was designed to address some of these requirements. The first major requirement is centered around the ability to have multiple visual perspectives of the same dataset. In SynVisio, we address this by two basic forms of visual representation available by default on the dashboard, and each of them focuses on one particular aspect of the dataset. First, we have the parallel plot where genomes are stacked horizontally and separated into chromosomes. Then, duplicated regions between the two genomes are connected through ribbons such that the size of the ribbon is based on the number of genes in that region and the color is based on the chromosome in the source genome. Following this, we have a corresponding dot plot where duplicated regions are shown as points on a two-dimensional matrix. Finally, we also have an extra scatter plot that visualizes additional information if needed about the duplicated regions, such as the extent of their similarity and the count of genes in each region. Another requirement is based on the ability to refine the visualizations dynamically through user interactions. This is achieved in SynVisio by letting users select and deselect chromosomes that they want to compare. So, a user can start by clicking on any particular chromosome and duplicated regions from that chromosome alone are then highlighted. This feature also works the other way around if you want to look at duplicated regions from a particular chromosome in the target genome. Following this, we also have a slider that users can use to refine the list of duplicated regions based on factors such as the extent of similarity to look for either close or distantly matched regions in terms of evolution. The third requirement is centered around the ability to investigate conservation at different genomic scales. So users can start with a visualization at the genomic level and then select a source chromosome followed by a target chromosome by simply clicking on them. This automatically updates the visualization to just show the duplicated regions in these two chromosomes alone. At this level, we use color for encoding the orientation of the duplicated region. So blue represents regular duplicated regions and red represents inversions in duplicated regions. Users can then explore further each region by clicking on it, which opens each of these genes in that region in a new panel. Here, because we opened an inverted duplicated region, the genes are all crossed up, but we can correct this by simply flipping the target genome. Thus, in this way, we can start at the genome level and explore the datasets all the way down to an individual gene. Finally, we also have the ability to explore genomic conservation beyond just a single source and target and compare multiple genomes. For this, SynVisio offers two variations of the basic parallel plot. The first is a height plot which uses a radial style layout. And the second is a tree plot where we stack parallel plots vertically and link the duplicated regions between them. Now, moving on to the evaluation of our system design, we use two basic forms of validation. For the first one, we interviewed 5 domain experts from our collaborating teams who used our system. All 5 experts stated that the tool helped them extensively in their research work and further confirmed that it was able to fill a critical gap by offering them a novel way to interact with the datasets. We summarized the detailed feedback from the experts in the form of case studies, which are explored in detail in our research paper. We also asked these experts to rate the usability of our system in exploring genomic conservation on a 5-point scale, from very bad to very good. And 4 out of 5 experts gave the system the highest rating of 5, and 1 researcher gave it a rating of 4. While this partially validates our design, to further evaluate our system, we made it publicly accessible as a web tool where researchers could upload their own datasets and monitor the web traffic for a period of 1 year to track system adoption rates. And our results showed that in this period, SynVisio had 150 plus users from around 18 different countries across the world interacting with the system in close to 250 plus sessions. Further, images generated by SynVisio have also been used in several publications, and our source code of the tool, made available through GitHub under a free license, has been adopted into several online genome databases. This usage of our system by researchers across the world in a wide variety of research projects shows that it is capable of supporting diverse datasets and user scenarios. In developing our system, we were able to surmise some design implications which we believe can also be used to inform other research in this design space. The first implication is that in visualizing complex genomic datasets, overloading of visual variables can be avoided by simply utilizing interaction techniques to bring up details on demand when necessary, or present them in a tiered manner that can be interactively navigated. Further, we show that circular visualizations that use circus-style representations, despite being popular for their aesthetics, aren't always the best choice for representation. Finally, visualization systems that use interaction techniques to switch context should have some form of support for revisitation, as users can often get lost during their exploration or want to revisit a particular form of the results again. To recap, our most important contribution in this research has been the design and evaluation of SynVisio, a web-based platform that lets researchers explore genomic conservation through novel interactive features. And the widespread adoption of our tool in a wide variety of research projects across the world validates our design. Thanks a lot for taking the time out to listen to this talk. If you have any further questions, please read the linked manuscript or drop us a mail.